Hello students, this is Geethanjali. Before going to replication, let us first understand the semi-conservative nature of DNA. So what does the semi-conservative nature of DNA mean? So let us take a DNA strand over here. Whenever replication takes place, the DNA strands temporarily detach, uncoil themselves, and a new strand of DNA is formed over the previous strand of DNA like this, and then we have our two copies of DNA formed. So you see the catch here. What happens is that whenever replication takes place, the new copy of DNA that is formed, in that one strand is from the previous cell itself, and this new strand is formed over it. So that is why we call this semi-conservative in nature. Now, how did we find about this nature? It was because of Michelson and Stahl's experiment that was performed in 1958. So, what they did is they took E. coli and they, for generations, grew it in a medium containing heavy nitrogen. Okay, and this heavy nitrogen uh, was in the source ammonium chloride, okay, heavy ammonium chloride. So as this was the only source of nitrogen, slowly the heavy nitrogen got incorporated in the DNA of the E. coli. So they took this E. coli containing heavy nitrogen and transferred it to a medium containing normal ammonium chloride. And they kept taking the DNA samples at definite intervals of time as the cells kept multiplying. So they took this DNA samples and they performed centrifugation in a cesium chloride density gradient. So we know that in centrifugation, the heavy stuff and the light stuff, they align themselves in a particular order, right? So wherever there's more gravitational force, the heavy stuff gets aligned. Whenever there's less gravitational force, the light stuff gets aligned. Keeping this in mind, the E. coli divides every 20 minutes. Okay, so they took the first generation after the 20 minutes of time interval, and they found out that on the density gradient, a hybrid band of DNA was obtained. So this indicates that it was a mixture of both heavy and light nitrogen. So it was in equal amount, that's why they formed a perfect hybrid. Okay, now again, additionally after 20 minutes, they again perform the same centrifugation, and this time they found out that there were two bands, and one band was hybrid and other one was light. So the explanation behind this is that the hybrid DNA that we find here, they separate, and only two strands of the heavy nitrogen are retained here too. Okay, as they were retained here, here also only two strands of heavy nitrogen are retained, right? And that is why the rest of it is just normal nitrogen, because the only source there we gave there was normal nitrogen, right? So that is why we obtain this results. So as they carefully observed these observations and came to a conclusion that DNA replicates itself in a semi-conservative manner. Now, another scientist in 1958, the same year, named Taylor, he and his colleagues, they used Vicia faba, okay, we commonly called as faba beans. They performed experiment by using radioactive thymidine to prove the same nature of DNA, that it replicates in a semi-conservative manner. We'll be doing replication of DNA. So replication takes place in the cytoplasm of the prokaryotes because we know that they do not have a well-defined nucleus. So we say it takes place in cytoplasm before cell division. Whereas in eukaryotes, the replication takes place in the nucleus, okay, before the G2 phase, that is the S phase. It takes place in the synthesis phase. So the first step here is assisted by an enzyme called helicase. So here we have a piece of DNA, okay, and this DNA has to unwind at particular places called ORI, that is the origin of a replication. So this helicase enzyme comes at the origin of replication sites and it unwinds the DNA. It basically uncoils the DNA. And this happens at specific places only, and these specific places are called origin of replication. To keep the DNA unwound, we have single strand binding protein. But we have another problem here. Whenever we unwind something in the middle and not at the end, supercoiling of the other parts is seen, that is in these parts. Okay, so to avoid that, we have DNA gyrase in prokaryotes and topoisomerase in eukaryotes. They prevent the supercoiling of the other ends of the DNA. So focusing on this part of the opening is called the replication fork. Now let us just assume that we have the 3' end here, 5' end here, 5' end over here, and 3' end over here. Now comes the real and the main enzyme, that is the DNA-dependent DNA polymerase. We call it DNA-dependent because it uses the previous strand of DNA to make the new strand of DNA. So what it basically does is that it uses a substrate, that is deoxyribonucleoside triphosphate, which also serves the purpose of providing energy for this process. So what we need to do is that we need to synthesize a new strand of DNA over here, like this, and then later it should separate. So that is what has to be done here, right? So this DNA, this deoxyribonucleoside triphosphate, it has 3 phosphate groups, right? So by breaking 2 phosphate groups, a lot of energy is obtained. So this energy is used by DNA polymerase to form the nucleic acid from the deoxyribonucleotide. So whenever we break these 2 phosphate groups, what is remaining is that deoxyribonucleotide, right? Because the deoxyribonucleotide has only 1 phosphate group, whereas this one has 3 phosphate groups. So after breaking 2 phosphate groups, we get the nucleotide. So this nucleotide is used by the DNA polymerase to form the nucleic acid here on the opposite strand. So here the DNA dependent DNA polymerase has a condition. it can only add nucleotides to 3' end. So because of this, it is only capable of functioning from 5' end to 3' end. So to provide this 3' end, we need another enzyme, that is the RNA primase. This RNA primase sets up primers which provide 3' end. So here, for this strand, we'll have a primer here. Okay, this will be our 5' end, then this will be our 3' end, and for this strand, our primer will be here. And this will be our 3' end, and this will be our 5' end. So now we know that the DNA polymerase enzyme will act here, and it will keep adding nucleotides according to the nucleotide here. Okay, so this DNA strand is called the template DNA strand, okay, because it acts as the template so that the DNA polymerase can put up the corresponding nucleotide with the complementary nitrogen base, right? So if it has T over here, it will put a nucleotide having A, adenosine. So it will keep moving on in this direction for this strand, and will keep moving on in this direction for this strand. Now, helicase again comes and it unwinds this part also. Okay, so after that, we'll be left with something like this, right? So we will have our 5' end over here and 3' end over here, 5' end over here and 3' end over here. So as we have just assumed that it has uncoiled a bit more part. So now we know that as substrate nucleotides have been added, our new 3' end will be formed over here. And in this case, our new 3' end will be formed here. Now for this strand, there is no problem. The DNA polymerase can continue its work with the 3' end over here. But for this strand, we have to place DNA over here. But we have 5' end over here. That is why we cannot proceed like this. So in this case, we need another primer which will provide a 3' end, and from this till the previous primer, the DNA polymerase will add the nucleotides. So you can see the DNA replication is continuous here. So here it is called non-continuous. Since this is continuous, we also call this leading strand, and we call this lagging strand. And these fragments that you see, we call them Okazaki fragments. Now this process is continued until the entire DNA has been replicated. Okay, so after that we have these Okazaki fragments, right? So to join these fragments, we have an enzyme called DNA ligase. This enzyme joins the fragments and makes it continuous at the end of the replication.